Okay, so uh, we had done up till this point, that is amniocentesis, that is mainly conducted. It is a kind of scan, a kind of CT scan where you are able to check for genetic diseases during the 14th or 15th week of gestation, to check for genetic diseases that may be developing in the baby fetus. Okay, so that is called the amniocentesis, which is done in the 14th or 15th week of gestation. Then Human Genome Project concluded that there are about 30,000 genes in human beings. Now, mesenchymal stem cells, these were discovered first in the bone marrow, capable of making bone, cartilage, and fat. So mesenchymal stem cells, they are derived from the bone marrow, right? Then induced pluripotent stem cells converted tissue specific stem cells into embryonic stem cells in lab. Induced pluripotent stem cells. All right, so induced pluripotent stem cells, so these are converted tissue specific stem cells, okay, into embryonic stem cells in the lab. So we are basically trying to convert tissue specific stem cells into embryonic stem cells, and this conversion is brought about in the lab. So they are mainly used in studying diseases, testing new drugs and therapies. So induced pluripotent stem cells are the cells that are actually these are stem cells, by the way, in the first place, but they are originally derived from the tissue, right? And then they are converted into embryonic stem cells in controlled conditions. So these are tissue specific stem cells which are converted into embryonic stem cells in laboratory. Then we have somatic adult stem cells, which are also known as tissue specific stem cells, and they can generate different cell types for the specific tissue or organ in which they live. So for example, like we take a stem cell of hair, and then we employ it in our hair, and then our hairs will regrow. So these are adult stem cells example. Okay, then we have embryonic stem cells. These are available 3 to 5 days after the egg is fertilized by the sperm, and these are derived from the inner cell mass of the blastocyst, and these are pluripotent. They can be differentiated into almost anything. So but the only drawback is there that the blastocyst then gets destroyed, and they are derived from the inner cell mass of the blastocyst. So that is what I'm trying to say, that there are basically 4 types of stem cells. The embryonic stem cells having the most potency of differentiation. Then somatic adult stem cells, well, they are derived from certain specific tissues, and they can differentiate into certain specific tissues only. And then we have induced pluripotent stem cells. So certain tissue stem cells are under controlled conditions in laboratory, artificially converted into embryonic stem cells. So that technique is used in studying diseases and testing new drugs and certain therapies. And these new induced cells are called as induced pluripotent stem cells. And then we have mesenchymal stem cells, which are the stem cells derived from the bone marrow. These are capable of making bone, cartilage and fat. Okay, so ये सब हो गया. Then इंजाज की बात करें, so that was the first cloned camel in Dubai, United Arab Emirates. And Lulu and Nana, these were the world's first genetically edited babies, I think by He Jiankui, by allegedly using CRISPR-Cas9 technology. So their name was Lulu and Nana. And then in 2010, Shrestha, that's the male cloned calf from somatic cells of an elite bull. So I think that was Shrestha. So somatic cells of elite bull were taken, and a male calf of a bull was made in 2010. Its name was Shrestha. Then 2010, Garima, second born through embryonic stem cells, which gave birth to a calf in 2013 called Mahima. Right. So 2009 mein Garima cloned calf. Again, it was a Garima cloned calf. 2009, Samrupa first cloned calf, but survived only 6 days. So you can remember the chronology, like these names. So Samrupa was the first cloned calf in 2009, and then came Garima, the second cloned calf, and after Garima came Mahima. So actually, Garima, which was a cloned calf, it lived for 2 years, and Garima, second born through embryonic stem cells, which gave birth to a calf in 2013 called Mahima. So all you need to remember is that the first one was Samrupa, the second was Garima, and the third was Mahima, and the fourth, or the 2010, that was the bull calf, that was the Shrestha, and it was a male calf, male cloned calf. Okay, then Lalji Singh is considered as the father of Indian DNA fingerprinting. Then we have Nobel Prize in 1962. Watson Crick को दिया गया था for double strand model of DNA along with Maurice. So remember these names for finding out the model of DNA. That's the double stranded model. It was given by Watson Crick and Maurice. Nobel Prize in 1962. Now, as we have already studied about the recombinant DNA technology, let's have a look at the steps involved in the recombinant DNA technology. You first isolate the gene of interest, and then you have to bind this gene of interest with a suitable vector, and then you have to introduce the vector into a suitable organism, and then you have to select newly formed recombinant cells with gene of interest. So once you introduce the vector into a suitable organism, you will witness newly formed recombinant cells with the gene of interest. Then you have to simply multiply those gene of interests. So I'm going to repeat that. The steps involved in the recombinant DNA technology: you have to first isolate the gene of interest, you have to put that into a vector, then you have to introduce the vector into a specified organism, a suitable organism. Now once you do that, your cells will start developing with the desired DNA, right? So these are the newly formed recombinant cells with the desired gene of interest. All you have to then do in the final step is multiply those gene of interests or those cells. So those are the steps involved in the recombinant DNA tech. Then restriction enzyme, I think Arbour and Hamilton were given the Nobel Prize for that. I think it was done in 1978. And so restriction enzyme, this is an enzyme which is used to cut DNA fragments, whereas DNA ligase is like a binder which is used to rejoin the DNA strands through phosphodiester bond. Just remember the name of this bond with which you are able to rejoin the DNA strands. So the enzyme or the restriction enzyme is used to cut the DNA strands, and then ligase enzyme is used to rejoin the DNA strands using a bond called phosphodiester bond. Then DNA fragments are inserted into a plasmid vector, creating a recombinant plasmid. This plasmid is introduced into bacteria which divide rapidly, thus rendering themselves as factories to copy DNA fragments in large quantities. So PBR 322 was first plasmid used in cloning Escherichia coli. You have to remember the name of this first plasmid in cloning the Escherichia coli. So I think this is the same as DNA recombinant technology. So यहाँ तो हमने simply पढ़ लिया, we isolate the gene of interest and then we put that gene of interest in a suitable vector. So that is what I'm saying. You have to the vector which we are talking about has to be in the form of a plasmid. So it has to be a plasmid vector. Okay, so this plasmid is introduced into the bacteria which divides rapidly, right? Thus rendering themselves as factories to copy DNA fragments in large quantities. So PBR 322 was the first such plasmid which was used in cloning the Escherichia coli. So यहाँ पे क्या कर रहे हो तुम, एक बार वेक्टर को फिर suitable organism में डाल रहे हो, that is nothing but a bacteria which has the potential to multiply. So it will multiply, and with the new DNA that was introduced in it, so selecting newly formed recombinant cells with the gene of interest and then multiplication of gene of interest. Okay, तो वही चीज़ यहाँ हो रही है. बेसिकली तुमने DNA लिया, उसको एक ऐसे वेक्टर में डाला which has to be in the form of a plasmid, and then that plasmid is nothing but a bacteria, I think. So DNA fragments, the desired DNA fragments, are inserted into a plasmid vector, creating a recombinant plasmid. This plasmid is introduced into the bacteria which divide rapidly. So the DNA fragments are attached with a plasmid vector, and then that plasmid vector has to go into a bacteria which has the potential to multiply. In case of cloning Escherichia coli, it was the PBR 322. Remember this name, it was PBR 322 plasmid. Then the plasmid which we are talking about, these are nothing but a small DNA molecule within a cell cytoplasm that is physically separated from chromosomal DNA, and can replicate independently. These are used as vectors. So these are that physically separated from the chromosomal DNA, right? So सेल साइटोप्लाज्म में तो होते हैं, बट ये क्रोमोसोमल डीएनए के इंडिपेंडेंट होते हैं, एंड these are themselves DNA molecules within a cell cytoplasm that are physically separated from the chromosomal DNA and can replicate independently. They can replicate independently. These are used as vectors. So वही मैंने अभी बताया है. Transgenic or GMO involves taking genes from one organism and putting them into another in ways that would not occur naturally. Bacteria are often used to grow I hate this dog. Bacteria are often used to grow and reproduce the desired genes as they grow and reproduce quickly and can be controlled easily in laboratory. Another ways of transferring genes is embryonic stem cell transfer, microinjection wherein the desired gene is inserted into the nucleus of egg cell and then egg is implanted in the surrogate mother. Okay, that is another way. So that's called as embryonic stem cell. So embryonic stem cell transfer is another way. Embryonic stem cell transfer is another way, where microinjection wherein the desired gene is inserted into the nucleus of egg cell and then egg is implanted into the surrogate mother. So that is embryonic stem cell transfer technique. But whereas this is यहाँ पे you have to take genes from one organism and put them into another in ways that would not occur naturally. So bacteria are often used to grow and reproduce the desired genes as they grow and reproduce quickly and can be controlled easily in the laboratory. Now, genetic engineering, manipulation, modification, recombination of DNA in order to modify the genome of an organism. ये सब कुछ तुम्हारा जेनेटिक इंजीनियरिंग में आता है. अब इसमें जेनेटिक लाइब्रेरी मेकिंग, ट्रांसजेनिक्स, थेरेप्यूटिक जीन थेरेपी, ये सब एप्लीकेशंस होते हैं. ठीक है. ओके, सो नाउ वी आर गोइंग टू लुक into some diseases of plants. Let's start from here. Okay, we'll start from here. Bunt. So what is bunt? I think it is a fungal disease. ठीक है. And whereas smut is a fungal disease of wheat, and bunt is I think it is a fungal disease caused by a fungus called Tilletia indica. And black rust, that is also, I guess, a fungal disease. What are we studying, by the way? So okay, I think we'll have to take help from AI. So what is bunt? Bunt is to hit bunt disease. So bunt disease is a fungal disease that affects wheat, drum wheat. No, it's not drum. It's durum wheat and triticale. It's also known as partial bunt, but bunt disease is caused by fungus Tilletia indica. All right, so it's a fungal disease, right? So whatever diseases are fungal disease, I will tell you. So bunt is a fungal disease caused by this fungus called Tilletia indica. And then black rust. What about black rust? So what about black rust? Black rust disease. So black stem rust, also known as cereal rust, is a disease caused by the fungus Puccinia graminis that affects many plants including wheat, oats, barley, rye and grasses. So black rust or black stem rust. So that is also a fungal disease. Okay, that is also a fungal disease. Where are we? Okay, so bunt is also a fungal disease. Black rust is also a fungal disease, typically in wheat, barley, etc. And smut is also a fungal disease of wheat. Now white rust occurs usually in Brassica family, that is mustard, cabbage, cauliflower and broccoli. You can remember these three: cabbage, cauliflower and broccoli. इनमें तुम्हारा white rust होता है. Albugo candida की वजह से ये होता है white rust. And symptom chlorosis of leaves हो जाता है symptom. देन अर्गॉट तुम्हारा सीरियल्स में होता है, स्पेशली बाजरा में अर्गॉट डिजीज़ होती है ड्यू टू फंगस कॉल्ड पुरपुरिया. एंड बाजरा फंगल डिजीज़ ग्रीन हेयर ड्यू टू स्लेरोस्पोरा ग्रामिनिकोला. ठीक है. तो मैंने तुम्हें बाजरा के मैंने तुम्हें दो डिजीज़ बताए, एक अर्गॉट और एक अर्गॉट बताया था और एक ग्रीन हेयर बताया था. ठीक है. तो ग्रीन हेयर अर्गॉट ये तुम्हारा बाजरा में होता है. ठीक है. वेरएज स्मट और बंट ये तुम्हारा वीट, बार्ली वगैरह में होता है. और वाइट रस्ट तुम्हारा होता है ये कैबेज, कॉलीफ्लावर, ब्रोकली वगैरह में ड्यू टू अ फंगस कॉल्ड एल्बुगो कैंडिडा. इसके अलावा वॉट और ब्लाइट ये प्रकार की कंडीशंस पोटैटो में होती है. ठीक है. पोटैटो में ये फंगल डिजीज़ वॉट और ब्लाइट के नाम से जाना जाता है, WART एंड BLIGHT, वॉट एंड ब्लाइट. देन टिक्का की बात करें, टिक्का इज़ अ फंगल डिजीज़ ऑफ ग्राउंडनट, एंड रेड रॉट ये फंगल डिजीज़ है शुगर केन का। और अरहर की जो दाल होती है उसमें भी विल्टिंग हो जाती है, दैट इज़ ऑल्सो कॉज ड्यू टू फंगस। देन देर आर सर्टेन बैक्टीरियल डिजीज़, तो अभी तक तो हम लोग फंगस डिजीज़ देख रहे थे। तो टिक्का में जैसे हम लोग ने देखा था, टिक्का इज़ अ डिजीज़ ऑफ ग्राउंडनट, रेड रॉट इज़ अ फंगल डिजीज़ ऑफ शुगर केन, एंड अरहर में विल्टिंग इज ड्यू टू फंगल डिजीज़, वॉट एंड ब्लाइट दैट इज़ ऑल्सो डिजीज़ ऑफ पोटैटो, फंगल डिजीज़। देन ग्रीन हेयर अर्गॉट, ये तुम्हारा ग्रीन हेयर तो होता है स्लेरोस्पोरा ग्रामिनिकोला से, और अर्गॉट होता है तुम्हारा पुरपुरिया से, बाजरा में, ठीक है। फिर कुछ बैक्टीरियल डिसीज़ हैं, जैसे मैंने लिख दिया है PCBC शॉर्ट फॉर्म में, दैट इज़ पोटैटो विल्ट, सिट्रस क्रैंकर एंड ब्लैक रॉट कैबेज एंड क्राउन गॉल। तो ये रट लो तो सही रहेगा। PCBC आर सम बैक्टीरियल डिसीजेस, तो पोटैटो विल्टिंग हो गई, और सिट्रस क्रैंकर इट्स अ बैक्टीरियल डिजीज़, पोटैटो विल्टिंग, सो PC हो गया। और फिर B से ब्लैक रॉट कैबेज हो गया तुम्हारा, और बैक्टीरियल से, और क्राउन गॉल ये भी तुम्हारा बैक्टीरियल डिजीज़ हो गया। फिर वायरल में टुंगरो डिजीज़ एक है, ये राइस में होता है, ग्रीन लीफ हॉपर से होता है ये, टुंगरो डिजीज़ राइस में होता है, वायरल डिजीज़ है ये। टोबैको मोज़ैक, येलो वेन मोज़ैक, ये ओकरा में होता है, ठीक है, ये भी वायरल डिजीज़ है, वाइट फ्लाई से होता है। तो ग्रीन लीफ हॉपर से टुंगरो डिजीज़ होता है राइस में, और टोबैको मोज़ैक डिजीज़ और येलो वेन मोज़ैक डिजीज़ ये भिंडी में होता है ड्यू टू वाइट फ्लाई, ठीक है। फिर पायरिला, दिस इज़ ऑल्सो अ वायरल डिजीज़ इन शुगर कैन ड्यू टू इंसेक्ट पायरिला लीफ हॉपर। एंड पाउडरी मिल्ड्यू, ये किस में होता है? पाउडरी मिल्ड्यू ये अनियन में होता है, मटर में होता है, और ग्रेप्स में होता है, ठीक है। तो बहुत ही फेमस है, मतलब पायरिला शुगर केन में होता है, एंड आई थिंक इट इज़ अ वायरल डिजीज़। दैन पाउडरी मिल्ड्यू किस में होता है? देखो, पाउडरी मिल्ड्यू मटर, अंगूर और अनियन में होता है, ठीक है। तो यही हम बताने की कोशिश कर रहे थे। बाकी बैक्टीरियल डिसीज़ में PCBC याद रखो ना, पोटैटो विल्टिंग, सिट्रस क्रैंकर, और ब्लैक रॉट कैबेज एंड क्राउन गॉल, सो दीज आर बैक्टीरियल डिजीज़। बाकी कुछ हैं वायरल डिजीज़, टुंगरो डिजीज़ राइस में ग्रीन लीफ हॉपर से, टोबैको मोज़ैक ये भी वायरल डिजीज़ है, येलो वेन मोज़ैक डिजीज़ ये भी वायरल डिजीज़ है, दोनों ही भिंडी में होता है और वाइट फ्लाई की वजह से होता है। पायरिला शुगर केन में होता है, पायरिला लीफ हॉपर की वजह से, और पाउडरी मिल्ड्यू ग्रेप्स, पीज एंड अनियन में होता है पाउडरी मिल्ड्यू, ठीक है। तो ये हमारे 2 पेज और हो गए आज के। फिर अब हम अगले में देखते हैं और डिजीज़ेस कुछ हैं, ये पेज नंबर 30 से।